Okay, let's talk about transcription. This is the first major stage in the process of gene expression. Now, transcription can be described as a process that is DNA directed, and it involves the synthesis of RNA. Now, transcription, or RNA synthesis, as it can be called, is the first stage of gene expression, like I stated, and it is catalyzed by the enzyme called RNA polymerase. RNA polymerase basically pries open the two strands of DNA, pries them apart, and then it starts to add RNA nucleotides using one of the strands of DNA, remember it's separated, as a template. So the RNA that is created is going to be complementary to the template strand. So that's the strand of DNA that is used as a template based on DNA base pairing rules. So RNA synthesis follows Chargaff's rules, except remember RNA has uracil instead of thymine. So whenever a thymine must be incorporated when it's complementary to adenine, a uracil is added instead. Now, the DNA sequence where the RNA polymerase attaches is called the promoter. So the promoter is a region on the DNA where RNA polymerase attaches to. Now, in bacteria, there's a sequence that signals the end of transcription, and this is called the terminator. Something else happens in eukaryotes, which we'll talk about later. And then the stretch of DNA that is transcribed is called the transcription unit. So the transcription unit is the unit that is going to be actually transcribed. So it's a section of DNA that is going to be transcribed into RNA or be used as a template to create RNA. So there are 3 stages of transcription. The 3 stages include initiation, elongation, and termination. So we're going to go through the little bit of details of each of those. So of course we're going to start with initiation of transcription starts when the RNA polymerase binds to the region on the DNA, which is called the promoter. So it's a region where the RNA polymerase binds. And the RNA polymerase binds with the help of what's called transcription factors. Transcription factors are just proteins which assist or they help regulate transcription, but they help basically attract the RNA polymerase to the promoter when that particular gene is meant to be transcribed. Now, within the promoter, there's called the start point. So this is the point where transcription is going to start, and it's just upstream of the transcriptional unit. Now, once the RNA polymerase binds to the DNA, and the transcription factors are also binding to the DNA, this is called the transcription initiation complex. Now, there's another region in the DNA that I must mention. It's called the Tata box. The Tata box is present in eukaryotic pieces of DNA, not in prokaryotic pieces of DNA. The Tata box is thought to be important in forming this transcription initiation complex. The Tata box is called so because you can see TA, well, it has alternating thymines and adenines in the DNA sequence. Here we can see a little bit of close-up of the eukaryotic transcriptional unit and the formation of the transcription initiation complex. So here we can see the promoter in eukaryotes includes the Tata box. You can see a bunch of T's or thymines and adenines inside the region that's called the Tata box, and again, it's part of the promoter. And then the start site is going to be just downstream of that. Now, here you can see a bunch of transcription factors. So these are basically transcription initiation factors, which are going to promote the binding of the RNA polymerase. So these transcription factors allow the regulation of transcription. So you don't always want to transcribe genes, just sometimes. And these are part of the signal that says, hey, this gene is going to be transcribed because it's needed by the cell. Now, elongation is the next step. Elongation involves the synthesis of, this is where the actual synthesis of RNA takes place. So RNA polymerase is an enzyme that actually has DNA unwinding capability. So it's able to separate the strands of the DNA and basically unwind it there, and then use that localized section that's separated as a template. So it uses one of those strands called the template strand as a template to synthesize a complementary strand of RNA. So you can see there's a complementary strand of RNA being synthesized and emerging out of the complex. So RNA polymerase works in the 5' to 3' direction. So it synthesizes RNA in the 5' to 3' direction. So you can see RNA here, we've got the template strand. The RNA is starting at the 5' end and then moving towards the 3' end. And you can see that synthesizing here. Now, this is a little bit of a close-up, so we can see this big bubble area is the RNA polymerase, and you can see it's separated the strands of DNA. And you can see the complementary base pairing. So here, there's an adenine, which is the next nucleotide in the DNA. Remember, the DNA is being used as a template. Well, if DNA, just in DNA synthesis, a thymine would be added next. However, we're synthesizing RNA, so a uracil is added next. So you can see that uracil being incorporated after the adenine, which binds to the thymine, which is in the DNA. So you can see the direction of transcription is moving to your right. And you can see the synthesis of the piece of RNA is starting from the 5' end and synthesizing in the 5' to 3' direction. Now, termination is the final step of transcription. And in eukaryotes, the RNA polymerase is going to transcribe what's called the polyadenylation signal sequence. So this is a sequence that is just upstream of the polyadenylation site. And the polyadenylation is basically a poly-A. So that means there's a string of adenines that will be added to the end of the RNA transcript. So the polyadenylation signal sequence signifies that the transcription unit is coming to an end and that the polyadenylation sequence is approaching, and that the RNA polymerase will eventually release the transcribed RNA about 10 to 35 nucleotides after the polyadenylation sequence. And here you can see a completed RNA transcript, and you can see the direction of transcription is going to be from the 5' to the 3' end. Now, termination of transcription in bacteria is a little bit different. Basically, bacteria is going to stop at the transcriptional terminator. Here, the messenger RNA can be transcribed without any further modification. However, in eukaryotes, the messenger RNA, okay, or really the RNA, it's not called the messenger RNA at this point, must be modified further. So we're going to talk about that in just a moment. So as I stated, in eukaryotes, the RNA that's synthesized from transcription is not completed RNA, so or messenger RNA, so it must be modified. So what it's actually called is the pre-messenger RNA. So this pre-messenger RNA must be modified in at least 3 different ways. This whole process of modification of the RNA is called RNA processing. Now, during RNA processing, both the ends of the RNA are going to be altered, and then regions in the middle of the RNA are also going to be altered. So let's talk about how that occurs. Okay, in terms of the ends of the RNA, really, it's not really called the messenger RNA, it's the pre-messenger RNA, right? So let's talk about both the 5' end and the 3' end. First, the 5' end. You can see at the 5' end of this pre-messenger RNA, there is a 5' cap that's added. So look, it's a G, but then with 3 phosphates attached to it. So these phosphates are not removed when the guanine is added to that 5' end. And you can see that they're basically inverted. So here the phosphates are directly attached to the 5' end of the pre-messenger RNA, and the guanine kind of hangs at the end. Normally it wouldn't be attached in this manner. However, this is the 5' cap, and this has a number of functions. However, it helps protect this end and help the ribosome recognize where to bind on the what would be the messenger RNA once it's done being modified. And then we have the 3' end. The 3' end is going to be where the poly A tail is going to be attached. So remember the polyadenylation signal. Okay, so that signal identifies the region where the RNA polymerase is told basically to stop transcribing the piece of messenger RNA, or RNA in this case, and it eventually stops. Well, at that end, okay, that's where the poly A tail is added. So what the poly A tail is just a bunch of adenines attached to the end. Now, both of these modifications to the 5' and the 3' end seem to facilitate or help the messenger RNA be exported from the nucleus to the cytoplasm, where it's going to be used as a template to create proteins. Now, they also protect the messenger RNA from enzymes that could hydrolyze those ends. Okay, so the 5' end has that 5' cap, which is the guanine cap, and then the 3' end has many, many adenines added to it. So if it does get hydrolyzed, none of the gene material will hopefully get hydrolyzed as well. And then again, the 5' end is thought to help the ribosomes attach to the messenger RNA at the correct location in order to produce proteins once it enters the cytoplasm. Now, there's one more modification that's discussed in your textbook, and it's called RNA splicing. Now, what RNA splicing, where it occurs is first in eukaryotic cells. Okay, so eukaryotes do the RNA splicing. So in the DNA, or ultimately transcribed in the RNA, are called introns and exons. The introns are going to be regions of nucleotides that do not code for protein. And then the exons are going to be the regions of nucleotides that do code for the proteins. So the introns are commonly called the non-coding regions, and the exons are often called the coding regions. And the coding regions are what is going to be expressed in the protein in some way. Now, sometimes introns must be removed. And the introns are going to be removed by this process, which I just mentioned, RNA splicing. So RNA splicing removes the introns and then joins the exons on either end of the introns to connect that to create the final messenger RNA. So let's take a look at what actually does this. So we can see that we have this pre-messenger RNA, the 5' cap and the 3' poly A tail has already been added. However, you can see there are several introns and exons in here. Okay, so let's say all of the introns are going to be spliced out. So once the introns are spliced out, okay, so those regions, those lighter regions that are labeled intron are going to be cut out, and then the exons on either side of those regions are going to be connected together. And this is the resulting final messenger RNA, which will be exported out of the nucleus and then be used to actually transcribe or translate proteins. Now, I want to mention a couple things that hadn't been mentioned before, and it's called the 3' UTR and the 3' UTR and the 5' UTR. UTR means untranslated regions. So this is another region that is not actually translated. However, part of it is expressed, I guess. It's not really the coding region. Okay, so you can see the coding region is going to be creating the protein, but the untranslated regions lie just outside of the coding region, but inside of the 5' cap and the 3' poly A tail. Now, these are usually regulatory regions which allow regulation of translation. Now let's talk about what actually does this splicing. It's called the spliceosome. The spliceosome is basically a complex of proteins and small nucleoriboproteins. So you can see we've got other proteins and then the small nucleoribonucleoproteins with some RNA in it. Okay, so this is another form of RNA. Now these are going to come together. Okay, and they're going to recognize, so there's some hydrogen bonding going on here. So there's what's called an intron-exon boundary. So the intron-exon boundary is a specific sequence that is recognized by these pieces of RNA that are associated with proteins within the spliceosome. They're going to recognize that, and then they're going to create this loop-like structure called the lariat. Okay, so this basically brings together the two exons that are on either side of this intron, which is eventually going to be cut out. So the spliceosome has some enzymatic activity. So it's going to cut the RNA at the boundaries between the introns and the exons, cut out that piece of intron. This intron will then be just degraded and then reused, or recycled really. And then you can see the two introns are connected together to create the final messenger RNA. So again, it's the spliceosome that actually does the RNA splicing. Now, what is the function of RNA splicing? Why is there introns in the segments of DNA and then ultimately RNA, and why is all this energy being invested in transcribing the introns if they're just going to be spliced out? Well, there's what's called alternative splicing. What this allows eukaryotic cells to do is to have one gene but be able to create multiple forms of a protein from that one gene by alternating or basically splicing out different combinations of the introns. So let's look at an overall gene. An overall gene is going to have domains. Okay, so these domains are going to be specific regions which are coded for ultimately in the DNA and then transcribed in the RNA and then translated once the protein is made. Now, exon shuffling is a process which allows new proteins to be formed. So this can create a situation where a domain from a protein or in the gene part of a protein can be moved to another protein. Okay, and then also I'm going to kind of elaborate on the alternative splicing as we look at this gene right here. So here we've got a gene in the DNA. And you can see in the gene we have the exons and introns. And remember, the introns are generally going to be those that are not translated, right? So they're going to be spliced out during RNA processing or RNA splicing. So remember the processes, we have transcription, RNA processing, and then translation we haven't talked about, but translation involves the production of the protein. Okay, so that it's using the final messenger RNA as kind of like a template, which carries a code, which was discussed in the first part. Remember the code, the triplet codons. Well, after translation, you can see the protein's made, and it's going to fold and it's going to create its three-dimensional shape. Remember, we talked about that in chapter 5. And we can see the different domains, right? So the different domains in this protein, there's 3 of them, and they correlate directly to each of the exons. So we have the exon 3, which is domain 3. We have exon 2, which is domain 2. And we have exon 1, which is domain 1. Well, this is assuming that both of these introns were removed. Well, let's say both the introns stayed. Let's say they were not spliced out. So there's going to be regions in between exon 1 and exon 2 and exon 2 and exon 3, which will make the protein larger, but it's coming from the same gene. So the protein will be slightly different in shape and larger, and it might have different function as well. So you can have alternating, you can change the combination of introns that are removed to create different forms of a protein from the same gene. This is called alternative splicing. Okay, so this is a way of creating new proteins with slightly new functions without adding DNA, right? So we've got this DNA, and it saves a lot of time. Okay, in summary, I described all of the steps of transcription, which involved initiation, elongation, and termination. Remember, there's some slight differences between prokaryotes and eukaryotes. And RNA processing, so that would be the next step during gene expression. Remember, RNA processing, in eukaryotes at least, involves the addition of the 5' cap and the 3' poly A tail and the splicing of introns. And remember, this RNA processing only occurs in eukaryotic cells, not prokaryotic cells. And then alternative splicing, I talked about, we've got the splicing of the introns, but what is the purpose and the function of alternative splicing, and how is it significant? All right, so thank you for listening. The next segment should be on translation.